mind control, mood regulation, memory implants. If I ask the majority of you what these things have in common today, you'd likely respond that it has something to do with sci-fi or fantasy. And you'd probably think that it's something that's going to happen in the next 20 or 40 years. And although you'd be sharing the opinion of a lot of people today, you would be wrong. Through the technique called optogenetics, all the things I mentioned are possible today. During my work at the NIH over the past 2 summers, I worked with a technique called optogenetics. As Kravitz, Anatol, and Lin showed in their 2012 paper, optogenetics is a technique that uses light-activated proteins to control neurons and alter behavior reversibly. So it's a powerful technique that uses light-activated proteins that are delivered to specific cell types in the brain, and they're used to excite or inhibit neurons and thus alter behavior. And today we are going to consider 3 points. The first is going to be how the technique works. So it uses a combination of genetic engineering and light technology. The second is going to be the fact that it has high spatial resolution, so it allows us to target a specific location in the brain. And the third is going to be about the temporal resolution of the technique, which means that it allows us to produce a response in a matter of seconds. So let's first focus on how the technique works. There's a protein called chanorodopsin. It's found in algae, and this protein actually responds to light. So when it's exposed to light, it causes an activation in whatever cell it's in. So the protein is inserted into a virus. The virus is injected into the brain, and it contains a secondary protein that will allow chanorodopsin to be expressed in only a specific cell type. This cell type will thus be the only cell type that is activated. And optic fiber implants are used to deliver the light. So when this light is delivered to the cell type, that cell type will be activated, and then the resulting behavior can be seen. And now let's go on to spatial resolution. So the technique allows a target of specific cell types, making it easy to relate a behavior with a specific region in the brain or a group of cells. This is unlike electrical stimulation that targets a wide area of brain tissue without specificity.